All right, welcome everyone. Thank you for coming today on this beautiful Saturday. I'm Emily Garner. I work at the MIT List Visual Arts Center in a neighboring building, and for those that are at the MIT community, building E15 or the Wiesner building. And we have a concurrent exhibition, which is why we have the joint program today as a nice collaboration with the MIT Museum, which is the building that we're situated in now. I just wanted to note that we'll provide a few program details, and I'll hand off the welcome introductions to our speakers today, to my colleague Ann Newman. But I also want to thank several people. We have an ASL interpreter and cart captioner on our live webcast, which is streaming currently now online for our virtual audiences. We also will be recording today's talk, and then we'll be putting it in post-production and posting. We will be doing a live Q&A at the conclusion of about like an hour moderated discussion, but with the post-production, we'll take the live Q&A off just so you feel comfortable asking your questions here today, and it won't be living on in perpetuity. Also with that, for hosting today's program, I have a few thank yous to give. One, a huge thank you to my colleague Carol Urbano, who is running around here, and she and I have done a lot of the program details and getting this program here, especially like only a month after this museum has opened. I also want to thank Artem, who is our MIT AV that's supporting our program today, and also the MIT Video Productions team, Kevin, who is doing our live webcast, and Miles and Clayton. And they also have been like organizing our CART and ASL services for today. The last part I would like to thank is our communications team, Leah Talatinian and the Office of the Arts, who supported publicity of this program, as well as at the List Center, Jameson Johnson, and then here at the MIT Museum, Martha Davis. And obviously we have our curatorial teams to thank, Selby Nimrod and Natalie Bell at the MIT List Visual Arts Center, who are also joining in the audience today, either one live and one in our virtual space. But then representing the exhibition today is Caroline Jones, who you'll hear a full introduction to. But I want to thank her for all of her programmatic contributions as well. And then we also have our wonderful other speakers here, but William Myers as the curator representative of the Gene Cultures. And then, of course, last but not least, but only because I'm turning it over to her, I want to say thank you to Ann Newman, who is the curator here at the MIT Museum, and she will formally provide the introductions. But thank you for joining us on this beautiful day.
So, welcome to our brand new museum. We've been open for about 32 days, I believe it is. MIT is precise, if nothing else. So we really appreciate this turnout and this sort of experimental practice space that we're trying to live in to allow and foster these conversations. I also wanted to, before I kind of give some context to the exhibition, thank the other colleagues that helped create the exhibition here. So William Myers, who's part of our panel, he was the curator for the bio art, but also Jacob Mons, who's with us in the audience, was the developer for the science and the technology that you see in conversation with this exhibition. So thank you for those of you who are here in this space, but also to those of you who are joining us virtually. I'm pleased to share that this first concurrent presentation of content between the List and the MIT Museum is both showcasing the fascinating works that are happening in the realm of bio-art and bio-design, and those are often intertwining practices, so we're here to kind of tease out the differentiation between some of those. So please come visit each of those exhibitions if you haven't had a chance to do so already. These exhibitions explore the perspectives of these artists and the researchers on this revolution in biotechnology and its power to alter life and nature. We're here with this distinguished panel to explore the emerging questions posed by the artists, questions of ethics, of the potential uses of gene editing, and of the place of humans and our relationship to other species and to the environments of the biosphere. In other words, can this biotechnological power allow us to redefine our relationships or our kinship to nature? And can we have a more collaborative and more equitable relationship with the technologists that propose these solutions to some of the challenges that we face? So I'm honored to present our panelists because they are the experts on the topics. and all of them are distinguished curators, educators, lecturers, and authors with an extensive list of credentials that I can't possibly have time to fit in today. But briefly, representing the list as part of the curatorial team for the Symbionts exhibition at the list is Carolyn Jones. She's a professor of history, theory, criticism in the Department of Architecture at MIT. She studies modern and contemporary art with a particular focus on its technological modes and its interfaces with physics, neuroscience, and biology. Her essays on modern and contemporary art have appeared in journals ranging from Art Forum to Critical Inquiry to Science in Context, and she's the author of several books and exhibition catalogs. Next is William Myers. He's the MIT guest curator for the BioArt and the Gene Cultures exhibition. He's the founding director for the Museum of 21st Century Design. Take a look at it online if you haven't seen it yet. William is an Amsterdam-based curator and teacher. He's worked in, lectured, or organized exhibitions in museums and learning institutions worldwide. He's the author of 2 influential volumes, one on bioart and one on biodesign. And his numerous exhibitions explore a range of topics, from the potential of biotechnology to revolutionize design, to the impacts of artificial intelligence on creative practice. And our moderator for the conversation today is Jens Hauser. He's a Paris and Copenhagen-based media studies scholar and art curator, focusing on the interactions between art and technology. He's currently a professor of art history at the Karlsruhe Institute of Technology, KIT. I've been told it's the MIT of a partner. He's a researcher at the University of Copenhagen's Medical Museum and at École Polytechnique Paris-Saclay, a senior postdoc researcher at the Medical University Vienna, and a distinguished affiliated faculty member at Michigan State University, where he co-directs the Bridge Artist in Residency program. So distinguished all of them. So thank you all for being here. And Jens, thank you for being willing to navigate this conversation. And I welcome all of you in the room to the discussion. Thank you.
Thanks, Anne, for the kind introduction. Thanks for inviting me here and to navigate not only through the conversation, but also to navigate across the ocean, according to a lot of issues that we have had with micro-performative agencies in the last years. I think we have to acknowledge that it's a kind of privilege to come back to travel, that we can meet again, that we can meet each other, human and non-human animals again, while the sound is sounding awkward. Okay, can we fix that? Thank you. So I tried to dress up in a way that it was both matching works in the exhibition, Symbionts and the Gene Cultures. So if I was confused with the picture, I wouldn't care. And there is also a nice exhibition element in the Symbionts exhibition that actually matches also very well. I spent 3 hours in each of the 2 exhibitions, and I have to say that I'm blown away by the quality and by the ambitions, by the challenges that they both pose. And being a curator myself, I also have to acknowledge and be very glad that I'm here for the first time meeting colleagues that I've heard of and that I admire the work of, such as Caroline and William. But it's actually today the first time that we currently and actually meet in person. So there's a kind of freshness, I think, in this discourses that we exchanged that was going to be involved, because we didn't prepare anything. So it's going to be fresh, hopefully a bit controversial, friendly, but I also, as a curator myself, and being also the author of publications and 30 shows of biotechnological art festivals and so on, I would like to address the issues of how these shows came together first, because very often people are not aware what kind of curatorial practice is behind these exhibitions. So before we start a debate about the conceptual issues, maybe both of you can just briefly introduce how the curatorial process and the call for artist works and so on worked. Maybe William would start. Are you
Sure.
Yeah.
Sure, and thank you for the question and the kind introduction. For us, the process was unfolding and evolving over time. Over the first few months, we held an open call inviting artists and designers to submit work that was about the changes in biotechnology over the last 20 years. With the idea that, or with the sort of the hunch from the very beginning that what we're witnessing in the unfolding of biotechnology is a bit like what maybe happened 20 years ago with the digital revolution, and how and why this time it can be different. So it's a long way of saying we had an open call, a number of submissions, and those were reviewed by a team of people that I was a part of. I like to think of curatorial work as a team sport. So there were other voices and perspectives brought to bear on selecting what works would be in the show, and then also what kinds of technologies we'd tell the story about alongside the artworks. And Jacob Mons, who was mentioned earlier, played a very key role in that process, as did a Rotterdam-based curator named Rawad Bakalini, who couldn't be here today. And over that course of selecting objects, then we began to talk about the spaces and how things would fit together and what kinds of, let's say, discourse or conversations, direct or indirect, could happen between them. And then at the same time that the installation was happening, this was also a construction site. So there was a pretty complex process that the team here managed through, and I think rather beautifully, to their credit. So that's a bit about our process. In the meantime, while this is happening, the curatorial team was writing texts to narrate what was going to be shown and fitting it into the larger discourse about biotech and art.
Did you coordinate each other in any way in the process?
We had a couple meetings.
We did, and it was, I think it was sort of early in COVID time.
Yeah.
So it was
COVID
video.
Tesseract, the COVID Tesseract, who knows when it was. But we were navigating partly our concepts to see whether we were going to be talking to each other or parallel play or whatever. And I think it kind of turned out to be parallel play, which is useful, and that's a bit what we're here to talk about. The very different approaches to biological materials in the two shows. There was one artist who shall be nameless who was on our list for a while thinking about that turned up on your list, and that.
Yeah,
So
I think it probably happened both ways. And
so they're not completely separate, but curators that I've known want to be first and want to be best. So of course there was some, creative competition. It was kind of, oh, you're doing a bio show, we're doing a bio show, right? And then we realized at the list anyway, with the fabulous curators there, Natalie Bell, Selby, director Paul Ha, we realized that this was really an opportunity to articulate our message and our approach quite crisply. Because I'm the one who's at MIT, and I've served at various wonderful moments on the MIT Collections Committee. So I remember even maybe 8 years ago, John Durant, who directs the MIT Museum, saying, wait, MIT Museum has it has thousands of slide rules. It has many, many engineering accomplishments. We don't yet have anything from the life science revolution. So John was, the director, right? So this is a bit of the backstory about how an exhibition might even be put on the calendar before someone brilliant like William is called in to play a role. So putting it on the calendar was the first step by the MIT Museum, as far as I know, to say, we need to have a voice and we need to articulate the story of MIT's own role in this revolution. Now, as an MIT humanist, the list is my privileged think tank, and I just want to say again, the team that William worked with, I also worked with a team. So I had this idea, I came to Paul Ha, he was losing his chief curator, he was getting another curator, he was postponing making a decision, so I was kind of in the antechamber, dreaming that the list would agree to commit its resources and its time and its galleries to this exhibition. So I was kind of pushing them, where you were solicited to help, right? So that's a difference there.
Sure, and as an outsider coming into an institution that I'm not familiar with, introduces all kinds of challenges.
Yeah.
I'll say that I think the first time coming here was as an advisor for the new building and how it would be designed and operated, and it was maybe a little bit of a job interview in disguise, because in the months that followed, I proposed doing work together, and it became like, a kind of mutual courtship, I think.
Oh, that's interesting. Okay, so I'm learning some stuff too. So the other context I want to make for the people who might not be here is that we are physically in this, fantastic new museum space, and around us is, of course, big pharma, right? So MIT has a very close relationship with these pharmaceutical bioengineering companies, startups, many of them started by MIT grads, right? So I consider part of my role at MIT to be a bit of the loyal opposition, to be a bit of the person who introduces a critical tone to that full-on positivist, we can create life, we can patent life, we can patent genes, right? I just want to be clear that I consider my role as, putting a bit of a wrench into that and say, slow down, let's think about that. So for me, art and this amazing new crop of bioartists that I was privileged to think with in the Symbionts show is a very different approach than the person that might want to make, mycelial leather, or might want to make, a giant salmon, right, 4 times as big as the regular salmon by inserting a pig gene, right? So definitely wanted to argue, in a sense, against gene culture, right? So I'm hoping we can get to these very significant differences. And part of my job is to shill the book. So we were extremely lucky to write and get a grant to make a gorgeous codex object. Even the shrink wrap is biodegradable. It's not really plastic, it's corn or something like that. So, this is design, right? We spent a lot of time with some amazing designers, omnivore, on this book, which, technically you can eat if you want. It has coffee waste paper, citrus waste paper, leather waste paper, right? So we can eat it and gain some nutritional value, but hopefully you'll read it first. Anyway, this is at the MIT bookstore, which is most definitely not the MIT Museum gift store. They have, again, separate territories, separate objectives, but I do hope people will take a gander at that. And you guys wrote beautiful brochures that anyone can take, because your show is quite complicated, right?
Well, it tries not to be. And I'm glad that you brought up this notion of having to combat a little bit the techno-optimism that is present in a lot of these settings, and particularly MIT, of course. So what we also try to do, and what I kind of view myself similar to how you describe, as someone that can introduce a critical voice into what is normally a narrative of, like, we can solve all the problems you can think of with technology.
Yeah.
When in fact we need to take a step back and consider who's controlling this, how is it implemented, is this widely or equitably distributed, this technology, and what are its consequences, and who gets to decide how it's implemented and where.
Maybe this is a way to already go a little bit into the title. The title was chosen, in agreement with everybody, so Syn versus Sim, and there's of course a deeper meaning in that, and the controversy already popped up a little bit. So Syn would be the kind of aim to synthesize, to be synthetic in relation to synthetic biology, all this nice gene editing technologies such as CRISPR-Cas and so on. And the Sim would stand more for the sympathetic, the symphony, to doing together. So there is this kind of literate kind of controversy already here. So I think I was brought in by the MIT because my practice was oscillating a little bit between the two worlds. I did myself curate exhibitions as early as in 2011 about art and synthetic biology. It was this synthetic, so synthetic and ethics mingled at the National History Museum in Vienna, where first went into the debate and having artists invited, 12 artists that were addressing hands-on different parts of what synthetic biology is now, beyond the kind of very cheesy and superficial equivalence that is open-made between synthetic biology and biobricks, and just to take it back from the genetic level also to origins of life research and proto-cell research to more material-based practices, and rather not just information-based practices. I've myself worked from national museums back to hacker spaces and punk spaces. So there's a rich range of practices that came up over the last 20 years. I personally curate exhibitions since a little bit more than 20 years. And in this 20 years, I think what was always key to me was to stage aliveness, and also back to art historical topoi, in how far the staging of aliveness was always entangled with developments of technology. And for example, if you like to see Byzantine hydraulic sculptures as a model, or the age of automata, or the mimicking of life as a social level, the intervention in real life that the Dadaists, Surrealists did, constructivism, and so on. So I think that for the last 100 years, and Jack Burnham is one of the philosophers of science and culture who put this up in his book Beyond Modern Sculpture, and addressing 2,500 years of how aliveness and staging of aliveness was always a driving force in the arts. And this is something interesting because first art was mimicking, imagining, then with the digital soft and hardware-based art, let's say simulate and actually manipulate the real living systems for real, so to come to aliveness in very literal sense. So I think there is a very interesting oscillation here at this round table between technoscientific media subverted, appropriated by artists from the molecular biology scale to ecology. And this is something, of course, that asks many questions in terms of collaborations also between art and science. And I think that we often have, and this is something I would like to ask you as well, what are these collaborations that artists then have with scientists from very different areas? And there's this asymmetric encounters, and I think that beyond this kind of mantra-like claimed art-science binary, which is very often present in discourse, it may be that the asymmetry also inherent in this institutional encounters generates disturbances, interferences. And I was just about to ask, so natural scientists often clearly distinguish themselves from engineers in a way comparable to artists distinguishing themselves from designers. Mm-hmm. So is the binary art and science, or are they finally on the same page? And maybe technoscience and design are closer to each other than, for example, and artists maybe and scientists in basic research are maybe more closer to each other. So maybe they think and they generally converge in their visions and their desire to reflect on how they know what they know. And knowing and feeling and translating aesthetically then how knowledge is being produced. And even the question of research then can transform in something else, to say you can, of course, research to find a solution, as technoscience does with a lot of these apparatuses that are in your show, and all this technoscientific gear that is, in fact, trying to find a solution for a question that is already posed. And artists, in turn, maybe understand the notion of research in a sense with the aim to generate new questions rather than find answers. So I want to, among the art and science residencies I have been helping to set up was the one in Germany, the Max Planck Institute. And that was now in class, knowledge links between art and science. And I was struck by one statement of an interview of a scientist collaborating with an artist at this point, saying that there is this blind spots that in this encounter can be revealed. And he said that one of the biggest temptations facing scientists today is the use of high-end technology instead of reason. Scientist saying that, not the artist. No? And if we are given a technological soul, we would rather just throw everything in a machine and then see what comes back. And a lot of artists, the scientist says, have noticed this back and forth with technology. While a new technology can help us seeing something differently, it can also obscure or distract the original attention. And I would like to challenge you both a little bit on that. Also to give it a start, because when I read through the two distinct descriptions of the show, William, in your gene cultures, a better revolution, you ask, so the entanglement between art and science, does it help producing a better revolution than the digital one that you just mentioned? And in how far innovation, creative and critical thinking can be fostered by braiding science and art narratives and stories of genetic research punctuated by projects from artists. So not to say illustrating, but punctuated, so to have a complete convergence. Well, when I saw the exhibition by Caroline,
And Natalie and Selby.
excuse me, I wanted to extend the credits to everybody. So you are focusing on what you consider new generation of practitioners are focusing on symbiosis, not on control, but with living, on organisms, not on processes that are just information-based, living entities, fungi, bacteria, instead of code-driven works, collaborative, independent works, not only as you write, using living materials, but welcoming them as teachers, partners in creation. So a question to get you started, and please do not hesitate to use examples of artworks to make your argument more tangible for the audience. Did you, in how far did you feel this, what Karen addressed as parallel play? In how far you see each other as parallel players as a part of this controversy that is very, tangible when you read the introduction to both of your catalogs. Do you want to start?
So maybe it's useful to talk about one of the works in your show. 3 collaborators, Christina Agapakis, who is trained as an artist and is locally based as the creative director of Ginkgo Bioworks. Sissel Tolas, who was trained as a chemist but practices as an artist in Berlin, and Daisy Ginsburg, who is a designer. And if I might just simply identify their project as a design project for me primarily, some of that is sociological. So it becomes exhibited at the Cooper Hewitt Museum of Design. Daisy has a very crisp design aesthetic and produces this as a kind of speculative fiction, and a very productive one, about an extinct smell that Sissel Tolas can conjure. And Sissel is a very interesting example because she was exhibited on her own as an artist at the List Visual Arts Center, right? So these worlds are not strictly apart. But we were aware when we were curating this exhibition of not going for that speculative design edge of things, which is very active right now, but in a sense, as Donna Haraway says, staying with the trouble, right? So take soil. Can we produce synthetic soil? Probably, right? But we wanted to stay with 2 powerful contemporary artists, Claire Pentecost, who's been attempting to shift, there is speculation and there is fabulation in the work, attempting to shift the US economy toward a soil-based economy and away from the abstraction of currency, right? So her designs for currency are made with soil, depicting soil workers from tardigrades to, farmers and theorists like, George Washington Carver, right? The workers of soil, the thinkers of soil, which he calls the teachers of Gaia. And then in the same gallery, Keon Williams for whom, as an African-American artist, soil is fraught because it is the place both of survivance and the place of enslaved labor, right? This is not easy to fit into a design package. It's not easy to fit into even the most provocative, speculative design project.
And it's worth mentioning that some of this distinction between art and design can be hard to make because some of the people that make these things will identify either as a designer or an artist. Sometimes they're trained in design, but their work is unmistakably artistic pieces.
Right.
And I'm happy you brought up this Resurrecting the Sublime is the name of the work from Daisy
The Extinct
Ginsburg
Smell, right.
and team. And I think at the heart of that work is something worth mentioning, which is to go through all this effort to resurrect a smell when the whole kind of resurrecting act is a bit absurd. That as species go extinct, the problem is not inventing technology to bring them back, but changing the way that we use technology so that extinction doesn't happen.
Yeah, this is perfect because the design piece of the extinction debate is like, let's just make a mammoth, which they're doing. Let's take an elephant and make it a mammoth by putting in genes, and then putting that embryo in the elephant and having a mammoth show up, right? Whereas the symbiosis argument is, I'm sorry, but we don't have the mammoth's ecosystem. There's nowhere for the mammoth to live. Mm-hmm. None of the plants the mammoth evolved to eat are still around, right? So the emphasis is very, very different. Design has a beautiful precision. There's a problem. We don't have a mammoth. Let's make a mammoth, right? I mean, I'm exaggerating. Whereas the artists we wanted to feature are all about sitting with the trouble and just, again, as Jens said, posing these questions.
It's interesting, maybe, to interject here, because in your show, William, there are 3 works of speculative design that precisely deal with extinct species. Mm-hmm. The chicken, the mermaid, and the smell. So,
well, chicken not yet extinct.
Well, the chicken is yet not extinct, but maybe you can elaborate on this because one of the first statements when Eduardo Katz started off with the notion of transgenic art back in the 90s, one of the claims he did, and was highly absurd and ironical, of course, was that, we are destroying biodiversity. Artists can help re-enrich biodiversity by doing artwork. And then this was, of course, very ironical and drawing attention to the fact that we are losing all these species and there is no technological fix for that. But you are actually having 3 extinction projects as speculative design in your show. What role do they play for you in this discourse?
Well, I just would underline what I said before, that the notion of extinction is one that I think in the hands of these particular artists and designers is an invitation to reflect on our human impact on the world, and the notion that works of art and design are maybe have a way towards improvement in their consideration of other species.
And a way of questioning. Yeah. I think in the Symbionts show, the work of Miriam Samoun has a lot to say to the chicken project, to the,
This is the project about the artificial pollinating.
So there are 3 parts to the piece, or the related parts of the piece. One, a kind of mural on the glass made of beeswax, a gift to us of the bees. And the artist is very engaged with honeybee collapse, which, the colony collapse, which is a symbiotic collapse. The honeybees have various insults to their system. They're actually non-unionized industrial farm workers. They're driven hundreds of miles to pollinate our fruits, our vegetables, et cetera. So they're very hard workers, and they're stressed. And we don't really know why their colonies are collapsing, but they are. So, okay, so there isn't a bee in her speculative project, but the film project is interspecies robot sex, because even when the bee pollinates the tree, that's an interspecies engagement, right? But now the fantasy is that maybe we need to roboticize that with tiny little drones made over at the Weiss Lab at Harvard, right? So she was an artist in place, working between labs here at MIT and at Harvard, making films of these projects. And, you know, that's a work that could have shown up in this show because she's intentionally pressuring the fantasy that a tiny drone will replace thousands of ecosystemic, you know, humming bees. So the European honeybee, which is the industrial farm worker, is one of hundreds of species, most of whom live in the ground, most of whom pollinate native species, right? So this is just her way of foregrounding the artifice of our contemporary relationship with these interspecies pollinators. Yeah, so.
Well, something I wanted to ask you about related to this and the notion of workers and underappreciated labor traces back to, I think it was a document that you shared with me a year and a half ago or something. It was like the first, or maybe it was more than 2 years ago, outlines of some of the thinking behind the Symbionts show and the research of Mary Margulis. And I might be pronouncing her last name incorrectly, but her vindication and her theory of endosymbiosis.
Yes.
And I thought this was such an awesome starting point. I didn't get to see the show in very much detail today, but I wonder in what ways, yeah, her thinking and her sort of, her indirectly or directly combating Darwinism and the notion of survival of the fittest and how that is played out in the world, how Mary's work informed some of your approach.
Yeah, Lynn Margulis.
Margulis,
who
yeah.
you're talking about, yeah. So grosso modo, there's a group of biologists who resisted the reductionism of gene Darwinism, if you will, right? Many of you might be familiar with the mainstream thing that, you know, once you had the DNA revolution, which itself discounted the work of female lab workers, taking the credit. I mean, you know, that's another story. Once Watson and Crick had the DNA helix, there was a rush to locate everything in that code, right? And this, broadly speaking, is called gene reductionism. And there were many, 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 particularly female and maybe later feminist biologists who resisted that move, right? Barbara McClintock, you know, people who insisted that epigenetic factors, what happens after you show up on this bacterial ball of a planet, right? Epigenetics was where the interesting things happen, right? So Margulis had an important voice in that, but it's extremely hard to get funding for symbiosis research to this day. So there was a biologist at Princeton who participated in a program funded by our Center for Art, Science, and Technology, who confessed to me that she was supposed to be writing a book about symbiosis, and it was very hard to get it published, right? Because the entire epistemology, if you will, I mean, you know, argue with me here, but the big epistemology is isolate, contain, don't cross-contaminate, you know, have it happen in the lab or it's not real, repeat it in the lab or it's not real, right? This is a huge thing. And most symbiotic relationships in the field cannot be replicated in the lab.
If you talk about artists as epistemological indicators, I think it's interesting. When we edited this volume here on microperformativity with performance research in the year 2020, we launched the call in 2019, before COVID, right? So we have 44 authors in this specific issue, talking about the notion of microperformativity beyond the scope of mesoscopic human intervention, so from the microscopic invisibility to the macroscopic incomprehensibility, and how far non-human agencies increasingly play a role in art, not just to be dominated and to be used as material, but as having their own agencies. And when we launched this call in 2019, nobody had the virus on the radar anymore. And I think 80% of our responses to the call for contributions was about bacteria and the microbiome and fungi. And the only one that we got actually to deal with viruses before COVID was even on the horizon was a paper by an Indian performance artist talking about traditional strategies in Indian Kerala area in how far communities have learned over the history and over the time to identify birds that bring issues and pathologies to the village. So to say they anticipate zoonosis.
Mm-hmm. Zoonotic diseases.
Exactly.
Right.
So this was the only transhistorical pattern that was anticipating the viral. Very interesting phenomenon. So very little works on genetics at the time. So of course we can anticipate that there's a kind of hype, and I would like to challenge you on this as well, because sometimes I had the impression that, well, artists are epistemological indicators, but they're also running behind. And jumping on the bus also by opportunism in a certain way, because there's a certain discourse around, and then sticking to the discourse, you know. And you have, I mean, I'm part of the same game. We just opened an exhibition, as you know, and we talked about Holobiont, Life is Other. Holobiont is the same.
One of the words coined by Lynn
Margulis, and also in Vienna at the exhibition that we just opened as well in October at the Angewandte Innovation Lab, the University of Applied Arts in Vienna. So it's about exactly these topics. So there's overlap, but we also self-critically question in how far that was a hype. And now bringing it back to the gender issue, I think that's interesting as well. In this team, we had 2 male and 2 female curators. In your team, you have 3 female curators. You already mentioned that.
Maybe.
I
mean,
predominantly.
Femme presenting.
Okay, very good issue. So just to not to draw to, of course, we are about to disentangle binaries, but my experience, and that's not a cliche, by really my experience in observing this field for 20 years, is that male curators often focus on genetics and synthesis, controlling nature, abstract universal codes, and female curators on epigenetics, as you say, cell and tissue culture, relational aesthetics of care, symbiotic, holiontic relationships, aesthetics of care.
I would like to locate this in culture, not hormones, but fair enough. I would say this is a cultural affiliation, a chosen affiliation. And certainly, but I will give a shout out to a male presenting scientist at MIT who was extremely important to our thinking because he is a geochemical symbiosis researcher. And he was the one who clarified for me just how challenging it is to research any symbiotic relationships the way science itself is configured. It's not easy to reconfigure science to do this. So he works with Penny Chisholm's lab. I don't know if she shows up in any of this, but she identified in the 90s a tiny phycoplankton. People knew that the chemistry was there, that something was making oxygen in the water, but no one had seen it. So you needed to get microscopes to a certain power before scientists could, as they say, isolate this organism. So Rogier Brockman works on detecting the symbiotic relationships as a virtue of their metabolites. Okay? This is not to take away from your analysis. I think your analysis is probably correct. But William has to own or disown the gene culture. So by adding culture, you're playing with the two meanings of culture.
Sure, but
the Petri dish culture and the big old messy.
Or the Winogradsky column culture.
Well,
the 2D and the 3D maybe as well.
I think underneath a lot of this that's so far not been recognized is that a lot of these works, I think in both shows, are responses to the upending of our shared notions of life, identity, and what's natural. And with the march of biotechnology, these things are really shifting, and they are fundamental, I think, blocks in understanding our world and our place in it. So in a similar way to the way that the surrealists were responding to mechanized war and the whole new terrain of the unconscious that was being illuminated by people like Freud and others, because Freud was problematic. I think bioartists today can be identified as offering those sort of responses. And I think this is a nice distinction to make in defining what bioart is to, I argue, not limit it to living media. That in fact, you could be a bioartist and you can take photographs. It's not necessary that you work with living things, but that you are commenting on or responding to these sort of upended notions that I just mentioned.
I think that's super helpful. And the artist Spela Petrich, whom Jens has worked with extensively, and in fact, I want to give Jens a shout out for this incredible documentation of her performance, which you commissioned and essentially made, or had made.
Thank you for that. I love Spela also, by the way.
Yeah. So this is an artist that is useful for us to talk about because Spela was a PhD biologist. And if I recall correctly, our discussions with her, she was quite dissatisfied with the scope of questions and work that she could do in biology, right? So this is another useful distinction to make. The scientist must write a short paper to prove a credibility to get another grant, to do the research for another short paper, right? The long form argument, as in a book, is a very rare piece of a scientific credential. The exceptions, prove this rule. So Lewis Thomas, Lives of a Cell. This was a highly Margulis-influenced popularizing book about how to think more capaciously at the unit of the cell and not be reductionist, right? So those are exceptions. So the scientist is usually given a very, very precise task, almost like a designer. Please, design this, solve this problem.
Yeah.
Be done, don't think further. And so Spela migrated, and Rogier hopes to stay as a research scientist because it allows him to think about the cosmos a little bit and not be worried about how he's going to fund 12 people in his lab for the next 5 years, right? It's a significant practice difference.
Sure. And one of the goals is always, of course, getting through peer review and just a set of considerations that are really unknown to the artist.
Right. And I maintain that research is a bit problematic as a term for artists because in the MIT culture, research comes with a deliverable. Yeah, sure, you can research, but you have a deadline and, show us your results. Mm-hmm. So research in a scientific and engineering context has to be scalable, has to be replicable, right? I tell the artists here, use that word with caution because what I wish for you is to have a space of speculation and a space of invention that is not tied to a directed outcome. So this might be a good challenge to think with whether how do these great projects happen when they are so clearly not giving us a new widget? How do these design projects come to be? What are the funding structures? So science and art will always, in my lifetime, remain very different in their funding and reward structures. And so the binary is robust in that sense.
Spela Petrich is obviously a very good example. And those who happen to be in Munich next week, she will actually perform this copotoisis piece, really shading a shadow of herself over the duration of 2 days.
Yeah, and we have a slide that comes up here. This big green field with a shadow on it is currently in a film form, 18 hours, if you choose to sit with it.
Exactly.
And the performance itself might be 20 hours. Yeah.
So it's going to be on stage live at Munich Muffathalle next week between 15th and 20th of November if you're around in Munich. But also Spela is a very good example because she actually incorporates not only questions of co-corporality with the vegetative, with the plant world, but also with teaching AI to be less biased by teaching AI with plants. So there's this design aspect to it, and also the other work that Spela Petrich is showing at the show that is coming up, Green Degrowth in Munich, is that she's showing also the sex toys for plants that you probably know as well.
That sounds great.
So how to help the vegetal others to have pleasure and to help them reproduce in a very pleasant way.
Although, of course, the very concept of pleasure is a mystery where plants are concerned.
Yes, and I think some of what we're talking about maybe gets to a little bit answering the question that you posed about how things are funded and supported. It seems to me in my years being exposed to some of this content that those works which can manage to speak on many levels to many types of audiences are the kind that get the most support. Like Spela, of course, is a great example because her work is all grounded in this educations that she's had in art school and in doing her PhD in biology. At the same time, she can step out of it a bit and name her projects things like sex toys for plants, and understand that that will get a giggle But if you dig layers and layers, there's much more to it. And that, brings up something else for me that I wanted to mention, that often it's the case with shows like this one, where over the process of curating, you ask the participants, the artists or the designers, to submit a text to describe what they did. And more often than not, I see these texts and I say, oh gosh, they're so talented, I wish they could write as well. But I've come to understand that that's really, that's my job, right? I need to get the description of it into words that are make sense, they're digestible, and they honor the complexity of the work. And I've also concluded that this is sometimes why they become an artist in the first place. If they could write really well, they would be doing that, but their expression is coming in another form, whether it's choreography, photography, dance, and so on. And this also, I mention it because it points to something that I think we've both hinted at in the written work that we've done accompanying the shows and in some of this conversation, which is that we really lack the sufficient metaphors and ways of description of what's happening in the sciences. Just to call DNA code, it is correct, but as you hinted that with epigenetics and with all the complications of life and all the stimulus that you have around you and how that comes out in gene expression, it's nonlinear, and it's not sufficient to think of it as a code like computer code or Morse code or what have you, that it's multidimensional and complex beyond what's graspable by the mind.
And
it's interesting in regards to the text that you wrote as an introduction for this book, that there's a genealogy.
Sorry, here's Stella.
That's the work.
This is the work, or the documentation.
The documentation. Yeah. In terms of history, it's interesting that you wrote this in a brilliant way in your introduction, that of course we had this 90s where the media arts were fostering a kind of confusion between the genetic code coming from cybernetics, all what Lily Kay has been writing brilliantly about, all this confusion also in metaphors, right? And that we have it here at the MIT, the Alexander Rich Lab, and Gamow as a mathematician playing a role, and Jacobson, the linguist, having roundtable discussions with François Jacob, the geneticist, and to talk about genetics as a language in a very structuralist way, before it gave way to post-structuralist analysis as well that questioned the code and the materiality of it. So I just wanted to point to the actuality of your show, because you come back to the genetic code in a different way, because maybe in terms of media theory, it fulfills a cycle, right? It was in Joe Davis' works, we see that in the beginning from the 1980s, it's to use DNA in prokaryotes as storage media, to store, and then to store extra biological information in biological media. And then, of course, with the time, this epigenetic thinking take over, and there's a Human Genome Project being superseded by the Human Microbiome Project, and then there's CRISPR-Cas. So genetics is back because there is this storing, transmitting, snipping in, infiltrating, injecting, erasing information, and now what we would able only be to read, we are able to write, synthesis and gene editing. So the actuality, I think, of your experiment relies on this loop that is being looped through CRISPR-Cas technology, because what you could store, transmit, is now being able to be edited. Would you agree that this is the new fashion about genetic technologies, that it's due mainly to the editing?
It's a new revolution, yet again.
In general, I would agree, but I think, again, we lack the metaphors to really describe it well. So when I hear about CRISPR-Cas9 and you think of editing, I think the most vibrant comparison that I can remember that came through in the beginning years of that research is that we had somehow invented the similarity to Microsoft Word, how we have a find and replace function, that we could do that now with genetic material. And maybe on a practical level, some of that is true, but it's such a, I don't have the words for it, right? We still rely on these sort of clumsy metaphors, and we tend to gravitate towards describing things in an oversimplified way, and with the supposition of linearity, that there's a text and there's a beginning and there's an end and there's a middle, when with genetic material and expression, it's really not the case.
I'm getting
So just
some
take the,
signs that
yeah, we should go to questions, but I just want to remind folks about the phrase junk DNA.
Oh yeah, very interesting.
So this is a great example of our tendency to, if we don't know about something, our tendency to assume it doesn't matter. So our metaphors rule us because they, as you pointed out in the essay of yours I read last night, they allow us to simplify our thought. They exclude many, many things. Our metaphors make it tidy and efficient to think. But junk DNA turned out to be everything about turning on and off, turning, light switch.
Yeah,
metaphor,
binary, yeah.
Right? Binary again. So these are, life is extremely complex. And the basic message of Symbionts is really to be curious and try and understand these operations that are announced as major breakthroughs, which are really advanced forms of animal husbandry, right? We grow things by making a broth and seeing if they'll show up and seeing if they'll replicate, right? We actually have very little capacity to invent life. We coax life into new forms.
François Jacob said evolution is a tinkerer, it's not an engineer. Evolution is tinkering, this famous article that he wrote. But I also would like to introduce maybe the notion that Thomas Feuerstein, an Austrian artist I'm working with a lot, working a lot with algae, says that we have to shift from metaphors to metabols. Mm-hmm. That means to read the actual metabolic process as if it was a language and to give to acknowledge what we have to read in the physical media and the materiality of the term. That brings me back to these two brochures, these brilliant brochures, one about symbionts and the other about gene cultures, and both surprisingly list extremely precise hints to philosophical resources beyond the actual work descriptions. But I was stuck in, especially your brochure, William, that you only list the artworks, while the half of the show is about scientific apparatuses and all the research fields that are involved, and that you have a 50-50 kind of relationship between artist and scientific discourse in the show. Why did you actually choose to not include the scientific discourse and only to make a brochure of the artist? Is art then playing such an important role, or why did you make a...
It could be that the stories about the art are just a little bit more engaging. But I don't think it was a completely deliberate decision. I think it was a matter of a number of factors, including that there were several exhibitions opening at once here. And some of the content, I think, was moved and borrowed at a late phase, so didn't allow for maybe the complete story of all the content.
Mm-hmm. Do you miss the scientific part, or would you have liked to include it, or is it indeed a personal decision as a curator to say, I would like to highlight the art as being this epistemological indicator in the light of all this techno-scientific gear that is up there, and not as a trade show, but something that is contextualizing within the realm of techno-scientific innovation and economy, of course?
That's a fair point, and I think to tell the most complete story, or to experience it most immersively, you have to see it yourself. And going to visit the galleries is the best way to do that.
If I could interrupt, I could take the pressure off of William, because it wasn't solely his decision. I don't know if I've lost. But I think we have, the museum is new, as it was said, and we're intentionally beginning to step into a space of using artworks to critique the technology that is happening all around us, to make that more apparent to our visitors who might be coming in and not expecting that as a continuum or a thread of thought or an area of exploration that we're pursuing. So it was our attempt to make that a little bit more explicit.
Should we open the floor for questions for the audience?
So thank you for a fascinating conversation that kind of expands our thinking about the porosity between art and science, some of the limitations. I loved the concept of the necessity of noise and how we play into those systems of noise throughout. So thank you for your participation, your questions. The exhibition at the List is open until 6 p.m. tonight. The museum here till 5 p.m. So with your minds fresh, on fire with some of these thoughts, please come through, take a look, and we welcome any feedback long term. So thank you to the panelists for your incredible work creating these exhibitions for us and for this fantastic conversation. Really appreciate it.
That was great, Jens. Thank you.
Yeah, thanks.